And now we will listen Tomasz Zajkowski from Polish Astrobiology Society and USRA, and he will tell us something about hunting on prions, ancient prions. The microphone is yours, Tomek.
Thank you very much. Great. So hello, everyone. My name is Tomasz Zajkowski. I'm a neurobiologist in training, but I study the role of amyloids and prions in the origin of life and early evolution. And today I will be talking about prions specifically and how we found them in the domain archaea. So I will start my talk by explaining what prions are. And when talking about prions, it's probably best to start with mad cow disease, because the term prion became recognizable by the general public because of the disease. But during this talk, I'd like to show you that prions don't have to be bad, but also they're very common in nature. So prions attracted global attention at the end of the last century when an epidemic of prion disease broke out in Great Britain. This epidemic was very unusual because it could spread between species, so precisely from cows to humans. And so far, there were 2 Nobel Prizes awarded for the study of prions, one for discovery of prion disease, and second for identifying the actual infectious agent as something even smaller than a virus, a protein. And this protein was given a name prion protein. Scientific community was reluctant to accept the discovery of infectious agent made only of protein. And prions are still the only infectious agent that has no nucleic acid component. And this difference makes prion replication fundamentally different from all biological processes. The fact that the prion can store biological information seems to be contradicting our understanding of the information transfer in biological system, so-called central dogma of molecular biology. But this uniqueness also promises amazing discoveries in the fundamentals of how the biology could work. Only now we begin to understand how important this phenomenon is in normal biology. So the most important discovery of the last 20 years or so is realization that prions are common in nature. First, thanks to the introduction of the concept of prions in the medicine, it became possible to start explaining very strange patterns of inheritance in yeast, followed by the discovery of many new proteins in very different species that behave similar to prions. So far, I told you how how prions were discovered and when you can find them, but I haven't told you yet what they actually are. So there is a very important distinction between prion itself and the prion protein. So prion proteins can acquire more than one fold, and one of these folds can aggregate, forming a prion. So the prion is not the protein itself, but the aggregate that can be inherited or transmitted to another cell or organism. So this animation shows formation of the original prion that causes mad cow disease. So at the beginning, the protein changes its secondary structure, increasing the content of beta sheet. And this misfolded protein is now interacting with other copies of the same protein. And it changes the conformation of the other copies to a similar one that it had. So the process repeats indefinitely, and while it continues, it builds up a higher order structure. It grows in one dimension and forms fiber. And such a fiber you can see on the right side of the slide. And these fibers are called amyloids. And most known prions acquire the form of amyloid. This is why the prion may also be seen as subtype of amyloid. And importantly, amyloids that are built out of the same protein can take a little bit different shapes. And the shapes depend on conformation of the protein, the starting conformation that it acquired. And Interestingly, the different shape of the fiber has different impact on infectivity and ability to cross species barrier. So the conclusion is that the biological information is stored in the structure of amyloid itself. So the information is stored in the structure of the aggregate. And it is possible that you already heard the term amyloid in the context of disease, in which amyloid accumulates in brain or other tissues, in Alzheimer, Parkinson, Huntington, even diabetes. And importantly, not all amyloids are bad. For example, protein called MAVS, MAVS, is a key to our ability to fight certain viral infections. In cells that are infected, it aggregates, induces production of interferon that recruits macrophages. Other examples are amyloids that are used in storing peptide hormones or building structures like silk or spider web. And on the image on the right, you see the matrix formed by amyloid protein produced by E. coli, Kerley protein or CSGA. And amyloids are very stable, and that explains some of their functions, especially in formation of biofilm. And also few amyloids are known in archaea, and they form tubular sheets. So again, just structure outside of the cell. Okay, but what is the difference between amyloid and prions? I told you that prions could be a subtype of amyloid. So the formation of a functional amyloid, like those that help with resistance or like forming biofilm and so on, it is often associated with gaining function that directly radiates from the physical properties of the fiber. And prion formation is often associated with the loss of function. So another difference is that prion aggregation is reversible, so the prion protein can act as bidirectional switch, while those functional amyloids, when they aggregate, they stay as they are. And so how do you turn off a prion? So let's start with the fact that prion can show up spontaneously. It is just a matter of statistics when a protein with an amino acid sequence that permits beta sheet formation will eventually acquire an amyloid promoting fold. And prion can also be inherited from like cytoplasmically, or can be picked up from environment and start turning other copies of the protein that builds it into the ones that fit into the aggregate. When the aggregate is formed, it can be either disassembled by protein remodeling factors or can be locked in the mother cells during division. And in this case, and both cases, the offspring would be free for prion, but this is going to happen after several generations, usually hundreds or thousands of generations. So in yeast prion, before they aggregate, like prion proteins, before they aggregate, they tend to be involved in processes that regulate the flow of genetic information. So transcription, RNA processing, and translation. So consequently, when the protein, like a prion protein aggregates, it means when it forms prion, it usually loses its function. And by losing its function, it affects the way that information is expressed. In consequence, it changes the phenotype of the cell. So the changes in phenotype, depending on environmental conditions, can be both beneficial and detrimental. So prion can be seen as an added layer of variation that in some cases can give population a greater chance of surviving. And the picture shows one of the applications of prions called biological bed hedging, that is in the yeast, a process that maximizes survival of a population in changing environment. So some cells are more likely to survive than others because they had a prion. So these observations are rather new and they raise important questions. Why yeast use prions as a source of phenotypic diversity rather than using a common genetic variations or transcriptional noise, for example? Also, how widespread it is the phenomenon among other species in other domains of life? But the most interesting question for me is, how old is the prion? Or how old is this phenomenon of forming prions? So in recent years, we have witnessed shift in perspective and perception of prions from a rare infectious agent to common proteins with important functions. But could this mechanism be also very old? At the beginning of my project, when I was looking through the literature, I found that prions have so far been verified experimentally within eukaryotes and only 2 bacteria, but none in archaea. And with that little number of examples, it is impossible to draw conclusions about the phylogenetic relationships of different prions. So our priority should be to find more prions to be able to assess their evolutionary origins. And my idea caught the attention of evolutionary biologist Lynn Rothschild, who became my supervisor at NASA. And this is how I started my hunt for ancient prions, specifically in archaea, because none of them were known. Our first step was to create a list of candidate prions in archaea. It is possible because prions have distinct amino acid composition, and because enough prions have been verified in yeast, it is possible to use amino acid composition of these known prions to train machine learning algorithms, and the algorithm can be used to scan the whole UniProt database and select candidates to be tested in laboratory. So our bioinformatic search performed on more than 1,000 archaeal proteomes showed that 873 proteomes had at least 1 prion candidate. And on the tree, the blue lines indicate the proteomes with at least 1 candidate and shows that distribution of prion candidates in the phylogenetic tree of archaea is more or less uniform. Some of them have more prions per genome. We're still trying to figure out why. And next, we performed an analysis of the functions of those candidates based on gene ontology, the geoterms, and we found strong enrichment among proteins related to the regulation of transcription. And this observation is consistent with observations in other organisms and is probably related to the fact that prions work as epigenetic molecular switch, and they just this function is better expressed in this context.
Tomek, we have 3 minutes.
All right, thank you. So just my results now. Then we conducted a series of experiments that confirmed the prion properties of 16 selected candidates, and we tested 16 out of 16 8-formed amyloids, and 6 were able to carry information by recreating non-Mendelian patterns of inheritance. And here are some experimental details. First experiment, I used method in which the candidate prion protein was expressed in E. coli and exported. And E. coli was growing on Congo red media, and Congo red binds specifically amyloid, and this was our first level of experimental screen. Then the successful candidates went for the electron microscopy examination of morphology, and there I confirmed that 8 out of 16 can actually form visible fibers, so amyloid fibers. And then consequently, using several spectroscopic methods, we confirmed the kinetic of aggregation that is typical for amyloid, so it has some anti-autocatalytical behavior, and that they are reaching beta sheets. That is also typical for amyloids. And then I used the yeast-based assay to test if prion candidates can function as protein-based elements of inheritance or information transfer. For this purpose, I replaced aggregating fragments, so-called prion domain, of a well-studied yeast prion protein, SUP35, with the fragment of the candidate prion from archaea that I was studying, and showed that candidates tested can rescue the phenotype, the prion phenotype in yeast. And in this assay, 6 candidates were able to create the prion, which persisted for many generations. It was trackable because the prion allowed cells to grow on adenine-free media, and colonies obtained a characteristic white color. And in conclusion, our experiments demonstrated for the first time that in proteomes of archaea, one can find proteins that can form prions. So we filled this dot, and our observations completed the picture of prion distribution within the main branches of the evolutionary tree of life, and suggested that there might be continuity between first abiotic amyloids and the modern prions that act as regulators of numerous biochemical processes, and unfortunately sometimes contributing to pathological conditions such as prion disease in humans. And with that, I'd like to thank all my collaborators, acknowledge and Polish funding agency that make this work possible. So thank you so much.
Thank you, Tomek. Perfectly in time. We can start a short session of questions. First question is from Kamil. Can some similar mechanism as replication or influencing other relative structures be found in other prebiotic molecules, such as PAH?
I'm not sure how similar it is. And PHS, well, they're too small to replicate the structure. They would be the same. They would just aggregate, right? So they would aggregate. So I'm really not sure. I don't know much about PHSs.
Tomek, I have a question. What do you think? What is how big is chance that you will find these prions in archaea?
Well, so what we did, we confirmed that a lot of proteins in archaea, when you extract them and you work in a model organism that is in E. coli or in yeast, they behave like prions. So now the biggest problem and experimental problem is to actually show it in the actual archaea. Well, archaea are pretty complicated to grow, but right now I have 2 collaborations with people that are able to work with archaea, but because you not only have to grow it, you also have to be able to modify it, and that's not that easy as it is in bacteria. You have to, it much more resembles modifications in eukaryotic cells, so it's pretty complicated. Not many people can do that. So we are constrained pretty much to archaeal species that are modifiable, and fortunately, there's plenty of candidates in these. And one of my colleagues already overexpressed 8 candidates, and one of them showed the signs of aggregation. So we are hopeful that we will actually show a prion behavior in an actual archaea pretty soon, hopefully.
Okay, thanks. Next question from Alec. What is the doubling time for the archaean species that are modifiable?
Yeah, so methanogens, they double pretty quickly, maybe twice slower than E. coli, so not that terrible. But we have some extremophilic archaea that we're very interested in, and they double once a week.
Okay, but still, it's much faster than most of them. I know some archaeons from ice which are double in, I don't know, few years or even a few dozen of years.
Wow.
This is still much better. But
no one would fund me that long.
Yeah, this of the 8 that you tested, one matched your results in Yeast. Are the others still being looked at, or they just did not work similarly?
Well, actually, out of 16 tested, 6 of them were able to work in yeast. So they were just the phenotype that is characteristic for yeast prion was rescued in 6 of them. So pretty a lot. And yes, we have plenty of candidates. The next step is to look into evolutionary conserved proteins, no longer just archaea, but we will look specifically at proteins that are conserved in bacteria, eukaryotes, and archaea. So in this regard, if we confirm the prion behavior, we would be certain to say that it is a pretty old mechanism, that the oldest proteins on Earth do behave like that. And this is pretty interesting concept because, yeah, these are mostly proteins that interact with DNA. And we also think about it from a bottom-up approach. Some amyloids, not necessarily prions, can be formed abiotically. So we see that maybe there is a connection that those first prebiotic amyloids formed aggregates like prion-like aggregates, and then they just sustained till today, acquiring some functions.
Okay, the last question from Kinga. I wonder, is any factor who is known causing change of conformation from alpha to beta?
Yeah, in my opinion, it is just a statistics. Important fact, if you overexpress the protein, then the prion is more likely to show. So yeah, it just happens. If there are any chemical, yes, pH influences it. There have been some programs that can predict a behavior of the fold in different pHs. So that would be another concentration of salt, temperature, these things. But remember that beta sheet is the oldest possible conformation of a protein. So, and reversing from alpha to beta is pretty easy.
Okay, thank you, Tomek. We need to finish, but I still have the question about culturing of archaeons, but maybe I will ask you later. But anyway, we need to finish now.